thank you. It's a great pleasure to be here and to represent the northern fringes of California botany. What I want to cover today. Considering the occasion, I'll indulge a little bit in the past, my own history and how I got into California botany. I'll then talk introduction to plant genomes, which is what I've been working on for the past 5 or 6 years. I'll give then a little bit of background on how chloroplast genomes and plant genomes differ and how they have been utilized in plant systematics and what we're doing with this now. Then I'll talk just 2 examples of my recent research in the family of and in the genetic area. Then a very short example of some work at the species level in the area of vesca. And then finally a few words about the future. So I actually got my start in California botany, really in Israel. I grew up next door in Arizona. Well, let's just say I didn't get out very much. After high school, I went to Israel to volunteer on a kibbutz and ended up falling in love with their very rich, diverse Mediterranean flora. It was great preparation for the invasive species and weeds. Very rich flora. I ended up studying at Hebrew University. This woman, Clara Henn, the herbarium director, she took a chance on a young American with no experience or skills, and hired me to work in the herbarium as an undergraduate student. And I ended up staying in the herbarium for almost 5 years. At herbarium, I did a master's thesis and then some postmaster's work with 2 plant ecologists these are plant ecologists whose work was really based on extremely strong knowledge and experience with the local flora. And they really gave me a very good perspective. But as I started to think about my further studies and desires to continue working on Mediterranean plants, it was clear that promised land was California, because these are the people who my mentors in Israel talked about. These are the people who they worked with, who they found to study with, and were inspirations to their work. And people like who I eventually worked with, Bob Thorne and Sherman Hoff, at Centennial Botanical Garden. Herb Baker, who's worked in both plant reproductive biology as well as in evolution of weeds, were of great interest to me. And Dan Axelrod's work in paleobotany, biogeography, was an influence on them and my early interests. Peter Raven and his work on California biogeography, speciation, and of course like Rich Evans and his great contributions that I did have the honor of actually working with, again, Thorne and Proudquist, and did interact and meet all of these botanists during my graduate and then postdoctoral works here in California. But it was in 1986 when Tom Elias basically recruited me to come to Rancho Santa Ana. He was building up the graduate program there, and there was an opportunity to do studies in California Mediterranean comparisons. The bigger influence was Warren Riesberg, who was hired the first year I was at Rancho Santa Ana. So he deflected me from the monograph that I intended to write to plant molecular systematics. And was both Warren and Leslie Gottlieb, who sadly passed away last year, really were pushing me in a more of a genetics direction, going beyond the using molecular data for phylogeny, which they both thought was important, but they really thought that focusing in on a plant and studying at a genetic level was the most important. And it's taken me over 20 years to get to that point, but I'll show you, I think that now, with the new tools that are available to us, we can now do genetics on any native plant, any wild plant. We're not limited to a small group of organisms that have a huge effort in developing the so-called model systems or model plants. So I think that opening up of these genomic tools to do genetic studies in any group is a very important aspect of my own research and what I think is coming in the future. Okay, so as far as my plant history, I have worked on quite a few California species, and definitely my background in Israel in ecology and floristics has kept me someone who's always been more of a generalist than a specialist. For about 15 years, though, this genus, the pines, really dominated my research and ended up with a lot of great collaborations in Mexico, around the world. Had some fantastic students and associates. And really, I feel like in the last, when Tom invited me to give this talk last year, he said, I hope you'll talk about pines, one of my favorite groups. I had to disappoint him and said, well, no, really, my life has been taken over by this plant, strawberry. And I'm going to talk about strawberries and roses instead. So I hope they'll forgive me for that. So why strawberries? Why are strawberries suddenly what dominates my research? Well, it's the simple fact about, let's see, late 2010, a little over 2 years ago now, the genome sequence of a Fragaria was sequenced. Fragaria vesca subspecies. It's named Hawaii 4 because it was collected on Hawaii, but the subspecies is native to Europe and it was actually an escaped plant in Hawaii, a volcano where it was collected. This is a diploid plant. And as we'll see, working with diploid plants, which have smaller and simpler genomes than polyploid plants, is very advantageous. It is considered a putative progenitor of the cultivated strawberry. This was the genome project for strawberry was not funded by industry. They had no interest in it. I mean, despite the fact the founders were very bitter that the California strawberry industry gave them absolutely no money. All they cared about were alternatives to novel bromide. So, much of a do-it-yourself genome project, starting up a little bit of money from Florida, but enlisted people, and as it turned out, there were 13 co-authors on this paper from Oregon State University. It was actually the single largest institutional contributor to the project of the 70 or so authors. So it was a great start for me to cut my teeth in genomics, and I've continued with that. The paper where it was published in for this pretty plant on I think they so the chocolate genome was published in the same issue. So. And I will return to the cultivated plant a little bit in some of our very recent work that is shedding new light on the origin of the edible strawberry. Well, they're all edible, but the cultivated strawberry. But the genome itself is from the diploid vesca. So as of a month or so ago, about 40 plant genomes have been sequenced. These folks, James Schnabel, Eric Wax, actually both work here at Berkeley, but now are in Tucson, University of Arizona, have been keeping up on this. If you look at the names, you'll see mostly it's cultivated plants, crops, or a few weeds like Caryopsis. There's actually 3 plants I would say are real plants, native plants in western U.S. I think in some ways, probably people who have been following plant genomics, plant evolution genomics, are much more familiar with the role of mimulus and the work that John Willis has done over the past 20 years to develop it as a model system for studying evolution, speciation, hybridization in natural species. And then Scott Hodges, his work in column lines is really advanced Aquilegia. So strawberry is not nearly as well known, but it is the first of these 3 to be published. A lot of in genomics, the data is generally released to the public, often years in advance of the actual publication. You really need to enlist a lot of people to help get the job done of the genome publication, so you make it public and let people start using it. But so we're still, you can use the mimulus and the Aquilegia data that are available through John Genome Institute across the bay, but the strawberry is the first one that was published. Now what is motivating this increase in 40 plant genomes as of March 1st, and I would predict probably at this rate, a plant genome is being published every week or two. What really motivates that is new technology. And this chart shows one aspect of that. This is the cost to produce the megabase. That's 1 million base pairs of DNA. In about 2007, we had this dramatic drop that reduced it by several orders of magnitude, so from $1,000 for 1 million base pairs, now literally 10 cents to produce 1 million base pairs. Now the advantage is that you can't just say, okay, give me my 1,000 base pairs for a fraction of a penny. It comes in pieces of about 10 million base pairs at a time. When we started in 2007, it was 10 million base pairs at a shot, and now it's up to 20 billion base pairs at a time. So that's more of the cost. So we had to be creative in being able to make it economical by sequencing many plant samples at once to take full advantage of the technology. But the pricing is there, it's becoming even more accessible. This is the technology that is really responsible for it. A company established in Cambridge, England, and I would say to predict that David Bentley, one of the inventors of it, probably will be in line for a Nobel Prize in the next decade. It was bought up by a San Diego company, Illumina, that bought it up in 2008 and has since developed it. So just to show the scale of it, so this is the slide, which is basically the size of a standard glass microscope slide. It has 8 lanes. Each lane can contain one set of DNA samples, but you could actually put multiple samples in there if you use what's called an index or a barcode for each DNA. And then this tile, which is about a half a millimeter square, has this constellation of several hundreds of thousands of clusters. Each one of those is a single DNA molecule that will be sequenced. So when we started doing this in 2007, we would typically get about 30 base pairs at a time. So you get lots of DNA, but again, I forgot to mention, it's in tiny little pieces. With platforms that have been released in the last 6 months, we're now getting close to 40, 50, 500 base pairs of these millions or so sequence. So it's really making it much more accessible and easier to work with. Why is the size? What is the importance of the size? So if we think about the genome, like a strawberry, it's really like putting it together is like putting together a jigsaw puzzle. So if you have pieces that are relatively large, and actually in this scale this would be huge, it would be like million base pair pieces, it's trivial to assemble that and put it together and see what you have. If you have smaller pieces, on the order, in this case, would be hundreds of thousands of base pairs, you start seeing ones that are identical. And this is analogous to a genome which has lots of repetitive DNA, so the repeat regions would either have very small differences. You only see the differences if you're looking at hundreds or thousands of base pairs, so they can be hard to piece together, or actually impossible to accurately, unambiguously piece together when you have that. Now in reality though, we're actually dealing with this situation where we don't have a bug, we actually have even tinier pieces. This is getting closer to the actual size of sub 100 base pair pieces. Oh, and there's no picture. We're working from scratch. So in the jargon, we call this reference guided assembly, where we have a reference to work with, to start with, and that can be done, and we've exploited that, and I'll show you in a moment. But then if you don't have a reference, we call this de novo assembly. So these are the two major methodological approaches that are used to make sense of these tiny little jigsaw pieces of DNA. Okay. So one more analogy here. So what is the scale of a DNA? I said you can get millions of base pairs, but is millions of base pairs enough? Well, no, you really need to sequence each piece tens, even up to 100 times to have enough data to really accurately tile together this jigsaw puzzle. And so we actually, it's, so if you need to basically what's called coverage, like 50x or 100x coverage. So as a scale, if you look at the Jepson Manual, I made a very rough estimate that the Jepson Manual has about 8 million letters in it. So if you take 8 million letters, and you want to say, so what is the strawberry genome, that would be a stack of about 25 of those. And 25 copies would give you 200 million base pairs. So that's the amount of base pairs that we're working with in something the size of the strawberry genome. And this is about the height of Now, if you look at the pine genome, which I used to work on, it's about 300 times larger. It would take about 3,000 copies of the, it's more than 1,000 times larger. 3,000 copies. So that would be a stack up to about here for the pine genome. It was about just below the tower of the Transamerican pyramid. So this is really the real reason that I switched from pines to strawberries. Okay, so one more little bit of biology before looking at some of the data and some of my results. If when you look at a typical plant cell here from the moss Plagionium, you can see that every single cell in a plant has tens to hundreds of chloroplasts. And in each one of these chloroplasts, there could be, again, tens to even 100 plastomes, so little circular plastid genomes. Each one of those is about 150,000 base pairs, so again, about 30 pages of the Jepson manual, which is a great place to start. 30 pages is about a very user-friendly size to work with. So the reason this is important is that when you just take DNA from a plant and grind it up and look at it, you're going to get a relatively high proportion of chloroplast DNA in your total extract. And in our experience, it's anywhere from, well, the lowest is about 1%, but up to 10, even 15, even as high as 20%. So in doing the sequencing, just straight sequencing, you can basically do what we call skim the chloroplast off the top. You'll get you'll sequence the chloroplast many times over, whereas you'll relatively sequence a small, each nuclear gene will get sequenced much less frequency because it's present only in a single copy, hidden behind all these chloroplasts, in this case in this cell. Now the other thing to think about is that, again, the chloroplast genome, the genome is full of nuclear genes. In strawberry, we're looking at about 30,000 genes. And this is really the target you want to be getting at, is sequences from these individual nuclear genes. Interestingly enough, we have been getting a sample of the nuclear genome. I still remember having lunch with Bruce Baldwin at Davis in 1990 when we were both there. And he was excited about this new area of the ribosomal repeats. And I remember being skeptical. I remember Les Scott was extremely skeptical of this, but Bruce persisted and proved us all wrong. And the key to the ribosomal repeats is that they are, in this case from Arabidopsis, you can see that they're actually they comprise about 5% of a nuclear genome in this case. And because of the evolutionary processes of the repeats, they very quickly become homogeneous, so they're all identical. So really it's like it's only a single sample of the nuclear genome, but it can be very representative. So for the past 20 years now, these have been the bread and butter of molecular systematics, the chloroplast genome and the nuclear genome, and the ribosomal DNA for nuclear genome. And people have wanted to work on nuclear genes, but for a variety of technical reasons, working on nuclear genes one by one, it's a lot of time, a lot of effort. And one of the things that we wanted to do is develop approaches to get the nuclear genome more efficiently. This is just showing, again, our diagram for genome skimming. So sequencing depth, this refers to the fact, as I said before, that you need to sequence a genome many times, the equivalent many times. So, for example, the 200 million base pair chloroplast genome, if you wanted 100x coverage, you'd have to sequence 20 billion base pairs, which again, is feasible. One experiment cost you $2,000. But that would be enough to start to be able to get reference-guided disassembly of nuclear genes. You might need more to do de novo. But if you have relatively low sequencing depth, you'll get the high copy fraction of the genome. So you look at the ribosomal DNA, shown here, you can get that sequence, and using with very low coverage, less than really 1% of the genome size can give you the ribosomal DNA. And the same thing with the chloroplast. You don't need a lot. You need about 100 million base pairs of DNA to get to be able to assemble with the reference guided approach to chloroplast. Oh, and another important point is that because chloroplasts evolve relatively slowly, we have a template, we have a framework we can work with. Most plant borders, soon most plant technologies have chloroplast genomes, so these can be used very effectively as references. And the ribosomal, the main active parts of it are very highly conserved, so those can also be used as a template, and then you can work and you can get your internal transcript spacer, your external transcript spacer very efficiently using these genome skimming approaches. So this is a very useful approach that we've developed over the last few years. Now here's one example. A PhD student at Oregon State University, Wambui Nguna, published a phylogeny of Fragaria using relatively complete chloroplast genomes, and about half of her data came from the genome skimming approach. You can do this with PCR. You can take the polymerase chain reaction, and in her case, she did over 50 reactions per chloroplast. It's a huge amount of work. It's a bookkeeping nightmare, but you can do it that way. I wouldn't recommend it, but when she started her project in 2009, that was the best. That was the approach we were using it then. So this tree, what I wanted to show you is kind of a preview of some of the Fragaria phylogeny that I'll go into in more detail with our nuclear data. But it does show, with her results, we had very nice resolution across the genus. Some important findings was the geographic separation of the mostly East Asian clade. We actually identified Fragaria vesca subspecies bracteata, which is the Western North American subspecies of the widespread diploid that I mentioned, as the sister group to a clade of octoploid species. And these octoploids are the ancestors of cultivated strawberry. We did some molecular talk work based on really excellent calibrations from really excellent fossils, recently described by Melanie DeBoer from the Republic Formation in northern Washington State. She has these beautiful flowers, young fruit of Malaria and prunus. So we were able to take as the earliest split between peach, of course, prunus, and then Malaria is closer to the apple clade. So that would be us to make there's a 49 million year split between those two. And since we also had chloroplast genomes for those, we came up with these estimates that Fragaria is a relatively young genus, 1 to 4 million year old divergence, and the octoploids are even younger. At the time when we published this, I wasn't aware of a report of a fossil Fragaria akin from the Pliocene of Canada, and that was dated at 2.9 million years old. So that was kind of nice to see after the fact some independent confirmation of the age of Fragaria. Now, as I said before, though, even though we had over 100,000 base pairs for these individuals, it's only a single estimate of phylogeny. And people like Jeff Doyle have been saying, telling us for 20 years that if we're basing a phylogenetic analysis on a single locus, it's basically a single character phylogeny. And none of us would want to base our classification on a single character like flower color, and we don't. And so why are we doing that in longer systematics? And it's been impractical to bring in hundreds of characters, again, until very recently. But I just wanted to give one dramatic example where relying on a single character can tell you something interesting biologically, but it's not giving you the phylogeny. So in sugar pine, we sequenced in chloroplast sequences about 2,000 base pairs, a couple of your standard non-coding loci, and we found an interesting phenomenon in this analysis of the white pines. You can see here sugar pine is coming out in two very different clades. Here is coming out actually identical sequence to Pinus albicolus, as well as several of the Eastern Asian fine white pines. And then here, strongly supported as sister to a group of Western North American down to Mexican white pines. Now, sugar pine is remarkable. If you look at it, as far as I can tell, no taxonomic variety within this plant has ever been described. So which is pretty unusual for pines. Most of them do have various alternate names, synonyms, or so on. But sugar pine is so distinctive, so unlike any other pine, that there's never been any issue of synonymous. So there's never been any idea. I thought there might be two species working in there that haven't been caught. So what is actually going on here? So when we map out the distribution of these, you can see this very nice pattern that you can see the yellow circles are the sugar pine with what I call the southern chloroplast type, and then the blues have the northern. These triangles are western white pine, or the white bark pine, Pinus albicolus, and they have the identical type. So what we hypothesized happened is that sometime in the Pleistocene, there was a event of basically illegitimate pollination of a Pinus albicaulis pollen grain onto a Lambertiana ovule, and because the pollen is transferred through the plastid is transferred through the pollen in pines, in contrast to the maternal inheritance in most angiosperms, that somehow the chloroplast got through, and either through chance or perhaps through selection has become dominant and widespread in the northern part. And there's actually some very interesting dynamics at this interface. I didn't put the slide in here, but in that picture we show that actually at this interface here between north of Mount Lassen, you can actually find plants that have a mixed, they have their maternal plant is a southern haplotype, but their seeds have been pollinated. We can see this by looking at the chloroplast of the embryo. We can see they've been pollinated by the northern branches. Some very interesting dynamics that could make follow studies. So basically, in this case, something has happened, but using the chloroplast as a marker for phylogeny, or even worse for identification, could be misleading in this case. This is one of the incentives to go beyond single markers like the chloroplast. So how do we do this? Well, in working on strawberry, it seemed like the rosaceae would be a great place to explore methods to try to bring in a large sample of the nuclear genome into our studies. So this is the results of a consensus phylogeny put together, led by Dan Potter at UC Davis, published in 2007. It really brought in several independent research groups that had been working on rosaceae phylogeny. And you can see that there's about 11 or 12 terminals on this. These were all well resolved as monophyletic. Several of them corresponded to traditional tribes that they recognized. And there were also species like Lionothamnus, Philopendula, Rufus, that they did not place for unresolved in this phylogeny. So at this point, I realized that having access to the complete genome of apple, of peach, and strawberry, could we use an approach to sample nuclear genes from across the entire family? People have been using this, for example, to sample nuclear genes of poplars using the poplar genome, other Mimulus species using the Mimulus genome, but could we extend this to an entire family, in this case using nuclear genes? And because nuclear genes do evolve at a much faster clip than the chloroplast or the ribosome, the boundaries of the ribosomal spacers, you really, I'm not sure we could go beyond the family as far as having universal markers, but I thought a family level would be a good place to start if we could expand from the species and genus into the family of markers. So to do this, I basically targeted 257 genes. So this is how much we sampled. And when you, these are the genes that were sampled. And to get down to 250, basically this has about 1% of a little bit less than 1% of the 30,000 genes. To get down to this, I basically used some filtering criteria. The genes had to be only a single copy in strawberry and peach. And because apple is the apple clade itself is a fairly recent polyploid, or tetraploid, we expected 2 copies in apple. These genes on one level had to be fairly conservative, that we could be confident of their evolutionary relationships to their orthology, so there had to be less than 15% divergence across the genomes, and this got rid of a vast majority of them. And another even stricter filter, there had to be nothing else in the genome that was less than 10% similar, because the approach I'm going to show you in a moment is based on solution hybridization, and if there's less than 10% similarity, you would get cross-hybridization. So basically, going between these 2 filters, they had to be relatively, they had to be relatively isolated in each genome and still comparable across a family. It got us down to this 250 or so range. So in the end, the average locus gave us about 1,700 base pairs, and our focus was on the exons, and on average there were 5.3 exons for each one that we targeted, and the total sample that we targeted was about a little less than between 420 and 450,000 base pairs in these 3 genomes. So that's about 1% of the 45,000 base pair coding region or exome of these species. Just briefly, the methods that we use here, typically called target enrichment. and the process starts with taking your raw DNA, creating what's called a library, where you put some adapters on the end of it. In this terminology, they're calling this the pond. We're basically going to go on a fishing trip. We're going to try to fish these out. And what we fish these out with is with RNA. The RNA comes from, this is actually microarray technology. Allows you to synthesize these little tiny droplets of each oligo. It's almost like a laser printer inkjet printer to put a little bit of DNA on this microscope slide. You elute it off, and through some PCR, you do two things. One, you add a tag to it called biotin. This will basically function as our hook. And then you also convert it into RNA, because DNA binding is much more efficient than DNA binding. So you take those two, you put them together in a tube, and you then let it sit for a day and a half at 65 degrees. You open the warm temp in the warm bath for a day and a half. At the end, the baits have hooked onto, via the RNA, they hooked onto the matching DNA. And then we use the fact that we can then find, so if it's hooked, it has the biotin on it. So we can then use this little piece of a little bit of iron filament coated with streptavidin. And the streptavidin binds to the biotin in an antibody-antigen type reaction. So those bind, or those can be preferentially taken out. So we're basically fishing out of this complex library of the entire genome, just those pieces that match our targeted RNA. So we pull this out in elution, and then a little bit of PCR to get the numbers up, and then we use those and go to those into the next generation sequencing. Just so we're able to do this in these experiments, basically each one of these day and a half hybridizations we do in a tube with up to 24 individuals per hybridization. We can multiplex 48 to 96 individuals in one lane of sequencing. Again, that costs us, those two steps cost a little over, whatever they cost. If you're going to the 48 individuals, it's about $75 per individual from beginning to end. And if you can actually do 96 plants on one of those lanes, which we think is now feasible with the high throughput, you're getting down to about $50 per individual for all of the materials and costs. And again, this can give you, in our Rosacea experiment, we've been pretty conservative. We have 250 loci, but there's no reason for the same amount of sequencing capacity of probe synthesis. You can get 1,000 loci, and get between 500,000 to 2 million base pairs of sequence per individual. And a very nice feature of this, we discovered that the process is not very efficient. Only about 50% of the DNA that comes through in this final process is hybridized. The other 50% is just the background genome. But that nice thing about that gives us enough sequence that we can use our skimming approach to do the chloroplast genomes and the ribosomal arrays. So we've called this hybrid approach, this whole approach, HiveSeq, to describe what we're doing. And in two of our papers, we published the essentials of the method. Even though this was done before the commercial kit from a company called Microarray was available, which has made it, which is what I've used as I see. Okay. So again, just a reminder, this is the biology we're working with. Good resolution of these terminal clades, but fairly poor resolution of the backbone. And I'm going to focus in just on 3 groups of interest. But I want to remind me to say that that's one thing that's interesting about the Rosaceae. Every one of these clades is represented in Western North America. And in fact, with the exception of Philopendula, which is found, it's very narrow, there's a Philopendula occidentalis is a very narrow endemic in the North Coast Ranges of Oregon, which is an area that has very low endemism. It's just solid Douglas fir forests, but there's one case of an endemic, probably less than endemics in the whole North Coast Range of California. But we do have this Philopendula occidentalis, which fills in the complete coverage for Western North America of all of these lineages of the Rosaceae. So what did we find? So when we look at our 250,000 genes, we get, first of all, we get beautiful resolution of this. the 2 rubus and 2 rosa, that was just a comparison of working with only the Fragaria only probes, or including the Fragaria peach and apple probes, to make sure there wasn't some bias introduced by having these heterogeneous probes from different genomes coming in, and they were basically identical. Some of the things that we find, I should say everyone, but you only see one bootstrap here, because every other clade is supported by 100% bootstrap support. Okay. Anyway, one of these maybe morphologically is that okay, this is the number. This is interesting too. This is the number of genes that support each one of those. So the 257 genes, you can see some of these relationships are supported by the great majority, but there's others actually that only have a handful of genes. And this actually, anybody who's worked with nuclear genes knows that it's, again, very much, many of them have very poor resolution, or only resolve one small part of the tree. So I really do think you need hundreds of genes to be able to get confident resolution. Some nodes may only be supported by a handful of genes total, but the overall consensus was extremely robust and consistent. So one finding that was important is that strong overall bootstrap support, but again, only 5 genes support the rhizoidy plus the triodoidy. This is a relationship that has great morphological evidence. You can see here Dryas looks just like, a lot like any one of your typical rhizoidy strawberry-like things. The feathered achenes common in a lot of in things like Pelugia are found again. But it was not found in any of the previous molecular results. But bringing all these data together does start. So it's a big surprise when you start looking at 1% of the genome, results start looking a lot more like morphology. That's the take home message there. Okay, Lionophamnus, I'm going to go through this. Basically, based on these results, Lionophamnus is sister root to peach. I think Lionophamnus has been argued as a paleoendemic and from the Channel Islands of California. And I think, again, really interesting. This has not been resolved before, but to think of the implications geographically of Lionophamnus being sister to this very large radiation of prunus, which is found throughout the more temperate region, is a new result. And also, a question about the North America, there's an interest of where did the apple clade, which has over 1,000 species of apples, hawthorns, their relatives, where did that come from? Previous molecular phylogenetic results resolved Galenia, an eastern North American species, or sister to it. And that's a herbaceous plant. It doesn't look anything like an apple. It doesn't look like it. And it always seemed kind of puzzling to me. And we did not sample Galenia in our data yet, but I do think that result seems robust. So I think what happens this is what we resolved is a good support between this clade of 3 tribes, which are all Western North American, primarily xeric genera, the Adenostoma, Ulmaria, and Nebusia, as sister to the Malidi. The other contender was here in the Spiroidy, which is again much more of a mesic group. So I think this does kind of put the origin of the apple clade in xeric Western North America. And it's interesting, the parallelism that then what we're hypothesizing is that there was just one dispersal here to Eastern North America, Galenia, and a very parallel thinking pattern here in Nebusia, where Nebusia alabamensis somehow just got dispersed to Eastern North America. But Tony represents the more ancestral biology of this group. Okay, so I'm going to, Fragaria, they all look alike. When I started they do. This is across the diversity. They all look alike. The fruits are more distinctive. The fruits do a terrible job of preserving them in a herbarium, so we don't really know them that well. So when I went in to apply this method to Fragaria, I didn't come in with very high hopes. As it turned out, though, every one of the species that we sampled, 3 or more, was resolved as monophyletic. So these are bootstraps, not as high, but everything in color represents a species, and they're all monophyletic. I'm still shocked by that result. But it does give a lot of hope for to be able to really, maybe for the first time, truly able to use monophyletic as a criterion in assessing species implementation. Just one of the number of genes that support each one, and again, the same point, sometimes a very small number of genes support it. But what's cool, even if you only have 4 genes supporting this clade and 6 supporting this out of the 37, there's a beautiful geographic split between the Eastern North American European vesta and then the Western Brachyata Mexicana California. So we're going to probably, there's probably good reasons to call them 2 species. Really, its name would be California. Okay. Just Okay. I don't have time to explain all this. Phylogenetic networks are something that I know is going to predict we'll be seeing a lot more in the coming years. And they're a bit, I was not trained in how to interpret them, but phylogenetic network, we did this for the polyploids because we haven't actually obtained the 250 nuclear genes from the polyploids yet. But we do have data from a different experiment where we have over 6,000 single nucleotide polymorphisms. And that allowed us to reconstruct this network showing a sister group relationship between these two octopoid species, Gregaria virginiana and Chilolensis. So basically, this shows that they're supported by over 7,000 of these shared polymorphisms. And there's strong support for, there's a split here, so there's over 5,000 SNPs that are shared by Inume, Japanese species, and these relative to the rest. And then there's 1,200 that are shared by Brachyata and Virginiana Chilolensis shown here. Sorry, not 1,200, there's a wrong reference. There's 1,000 here. And then finally, if we bring in Vesca, over here, it adds another 800. So basically, I think there's some ancestral traits that are shared across it. So this allows us to suggest that one of the parents of these octoploids, well, clearly one of the parents was Inume. It probably generated two genomes. It's an octoploid. One of them was probably an ancestor of Vesca, perhaps with more of the polymorphism retained today in Brachyata. And we think there's a missing ancestor that we haven't found, but in my view, probably extinct. And I mentioned before that fossil, but here's the ancestors, and the timing puts it to the Pliocene when there was good connection across Thuringia. The fossil data is there. Inume is Japanese. Iterokensis is an octopod only found on one island on the Sakhalin chain, which are the Kuril archipelago. But it would suggest an origin of the octopod somewhere up here, with one relative over here, and then very widespread Chilolensis and Virginia here. And the cultivated strawberry, so it's from France. How to get to France? Well, Gregaria chilolensis was introduced to Europe from South America. It's an amphitropical species in the 1700s. Virginiana had been introduced from Montreal in the 1600s. And French gardeners discovered this in the mid-1700s. It was a very tasty nutmeg. What is less well known is that these two species hybridize naturally in the Northwest. They're actually extremely common in the Willamette Valley. And we have a paper coming out in the next Journal of American Journal of Botany where we'll go over this in detail. But this explains why the native strawberries in the Willamette Valley are so tasty, because they have the same genetic background as the cultivated strawberry. Am I out of time? Okay. The last thing I won't have time to go into this, but I just wanted to show that we're also very excited about using these methods to look at single genes, finding genes. So it's kind of the holy grail. This is what Les Gottlieb and Lauren Wiesberg wanted me to do 20 years ago. It's really becoming practical. Here we're looking at a trait of male sterility or female plants in these strawberries. And basically, this is showing us that we have strong support, strong evidence for a gene in here, linkage group 4, in a population from for this gynodation Brachyata. And we've actually, using the genome, we can actually take it down to a level where we've got only 350,000 base pairs that target and 57 candidate genes. So the limiting factor here is not the sequencing, but basically having enough progeny to find recombinations. So we're increasing our progeny from 100 to 1,000 to try to narrow in on the genes. So we're very close on that. Then the final result, we now look at a population in New Mexico, and it's a different chromosome, different location. One of the real frontiers for molecular biology. Molecular biologists are discovering diversity. There's as much molecular diversity as there is organismal diversity. And it's a big shock to them. A lot of role for evolutionary biologists and systematists to help them wade through the discovery of molecular diversity. Okay. Well, a couple take-home messages. Anyone can sequence a genome? Good questions that are a premium. Again, people, ecologists, evolutionary biologists, we have the good questions. And you can go a long way to help genomicists figure out what to do with the data. If you're just getting your feet wet, if you're a grad student, doing any kind of population level work today, you should be working with simple sequencing peaks or microsatellites. Very, very easy to get. Bar-class genome is a great place to start. The HiveSeq method, I think it has huge potential. And one aspect that I think is extremely exciting is that we can use the same set of markers, the same nuclear loci to look at phylogenetics, population genetics, and linkage mapping. They work at all three levels in the same markers. There's enough variation across these loci. You can do that with one experiment. I'm very excited and that's what we're developing here right now. And then again, just linking the phenotypes to genotypes is becoming feasible in wild plants. Oh, I should mention that in New Mexico, the experiment was done from a self-plant, a self-individual. In traditional genetics, you can't work with cells, not enough variation. But we had over 1,000 markers from the cell, which means you can go into the field, you can bag a plant, you can collect the seed from the cell and do this kind of thing. And final thing, of course, maybe thinking of you, but again, how are we going to analyze gigabytes of data, gigabases of data? I think if you think about Sanger sequencing, it never had much impact in what really matters in biology, of course, human health, human disease. And it was never really used very much, but there's a lot of interest in using, well, more than interest, there's definitely moving towards using genomics, next-generation sequencing in human health, human diagnostic. This company, 23andMe, is aiming to have 1 million customers by the end of the year who will know what a SNP is and be, analyzing it. So there's a lot more awareness, a lot more interest in making this broadly available. And I think that's going to benefit all of us who want to use the tools that are being developed to easily analyze it. I mean, if your medical doctor can understand it, of course we can too, right? And then you can do some things like if you get 1 million SNPs for $100 and find out the end of all the credit.